Hi, Year 11s and 12s. Today we're going to be looking at DNA profiling. This goes along with Unit 1.5 in your SASTA workbook. This PowerPoint is going to be split into 4 different videos for ease of understanding because there is a lot of different concepts we've got to look at. This is the first video looking at just what is DNA profiling. As per usual, you've got your need to know. As you can tell, there's a lot of need to know statements from the syllabus in this guide. I'm not going to go through all of these in the video. If you want to write these down, it's a good spot to pause the video and physically write them down. To start us off though, we're going to look at exactly what is DNA profiling, then we'll look at what a DNA profile physically is. So DNA profiling is when scientists study the genome of living things in order for them to determine the location and function of genes in the human body. It doesn't have to be human bodies, obviously we look at genomes of other animals too. But by doing this, it allows us to determine the genetic and evolutionary relationships between different organisms. It also helps us to identify if someone was at a crime scene. Only with further evidence could you solve the crime. So finding DNA at a crime scene is not enough to convict someone. That's an important thing to note because they try and trick you with questions in that instance. And lastly, to help identify gene mutations that will lead to the development of genetic diseases. So now that we know what DNA profiling is, what exactly is a DNA profile? Well, a DNA profile is the DNA-based pattern made up of different bands corresponding to the DNA fragments of different sizes. So we can see over here, we've got the different bands based on the different sizes of the DNA. If we were looking at this one and trying to determine which person was at the crime scene, and remember again, we're not saying which person is the criminal, and we can see that the victims ones are lined up here, so we can see the victim was most likely at the crime scene. But then we need to find out which other ones are there. Okay, and for that, we're most likely looking at suspect 2 because it shares the most similarities, if not identical, to the evidence or the DNA profile that's been found at the crime scene. Okay. So a series of bands corresponding to DNA fragments of size. We also call it a fingerprint, because like we can tell over here again, we've got the 3 different suspects and they all have their individual DNA profile. So like a fingerprint, the combination of bands in a DNA profile is unique to each person. Now when you're looking at that in detail, half of the bands are from your mother, half are from your father. So it also helps with paternity and maternity tests as well. You can see if the DNA combines to form that new profile. Before you create a DNA profile, and that's what we're going to talk about this week, is basically how we're going to create a DNA profile. You need to be able to extract the DNA from a cell or from the evidence that you've got. Now, you've done this already this year when we extracted DNA from a strawberry. Okay. DNA extraction, like I just said, it's the process of physically removing DNA from a cell. Generally, detergent and salt are added, and the reason for detergent and salt being added is to dissolve the cell membrane and allow us to get the DNA from that membrane. We then also add ethanol, so we did that with methylated spirits when we did the strawberries, and we precipitated the DNA so it all builds up together so we can easily extract it. This is the whole thing you did with the strawberries. So if you go back and have a look at the practical we did in class with the strawberries, that's how we extract DNA. Now I'm going to do the DNA extraction again this week, and we're actually going to look at what that DNA looks like once it's removed from the strawberry under the scanning electron microscope. We'll talk about that in class though.